Hello students, welcome to EPG Pathshala. I am Dr. Nilofer Khan from DPAS DRDO Delhi. Today I am going to talk about relation between membrane potential and cell characteristics, membrane impedance from paper Membrane Biophysics. And the objective of the talk are electrical properties of the cell membranes, conductance and capacitance, electrical potential across the cell membrane, membrane proteins, ionic basis of membrane potential, and membrane impedance. And I want to give some introduction about the topic. A cell, which is a fundamental unit of all biological life, is surrounded by a cell membrane, or it is a plasma membrane. It is also called lipid bilayer, and one of the typical feature of a cell is that it regulates the transport of electrically charged ions across the lipid bilayer. Therefore, the electrical potential across the cell membrane. The hydrophilic membrane bilayer is an impermeable barrier to inorganic ions, and it is a poor conductor of electricity. What are poor conductors? Poor conductors are those which have a very high resistance to electrical current. In this case is the ionic current. Ion channels act as ion conductors in the membrane. They established a conduction pathway into the membrane and lower its resistance. Properties of the cell membranes. All cells are enclosed by a plasma membrane. Most plasma membranes composed of approximately 50% lipid and 50% protein by weight, with the carbohydrates portion of the glycolipids and glycoproteins constituting 5 to 10% of the membrane mass. The lipids of the plasma membrane or bilayers are amphipathic, that is, their molecules have both polar head and non-polar tail. The polar regions are hydrophilic and the non-polar are hydrophobic. While lipids are the fundamental structural elements of the membrane, proteins are responsible for performing particular membrane functions. Jonathan Singer and Gert Nicholson in 1972 proposed the fluid mosaic model of the membrane structure, which is now generally established as the basic concept for the organization of all biological membranes. In principle, plasma membrane is an electrically non-conducting thin phospholipid bilayer of about 6 to 9 nanometers. There are also integral molecules with various functions. Considering electrical phenomena, two kinds of membrane proteins are most important, the ion channels and the pumps. One type of the membrane can be distinguished from the another type by the nature of the protein it contains. In neuronal membrane, there appear to be 5 functional types of membrane proteins. Electrical properties of the cell membranes. The electrical properties arise from the physical properties of the cell material and from the ion channels in the membrane. The cell membrane acts like a simple electrical circuit containing resistance and capacitance. Resistance, it is a measure of the difficulty with which current flows in an electrical circuit. Greater the difficulty, greater the resistance. It is abbreviated as R. The unit of resistance is ohm. Conductance, the inverse of resistance is called conductance, abbreviated capital G, an expression of the ease with which electrical current flows in a circuit. The standard unit of conductance is Siemens, S. Previously it was known as mho. Current is a flow of electrical charge, I, measured in coulombs per second or ampere. It is abbreviated as capital A. Voltage or potential difference is a separation of unlike charges in space. The greater the amount of charge separated, the larger the voltage, and the greater the tendency for the charges to flow towards each other. Voltage is always measured at one point with respect to another point. There cannot be a voltage at one point in space. The unit of voltage is volt, is abbreviated as capital V. Voltage, current and resistance are related to each other by Ohm's law, that is V is equal to IR. Ohm's law can be rewritten as I is equal to GV, where G is equal to 1 over R. In this form, the equation current is a direct function of both voltage and conductance. Conductance. The pure phospholipid bilayers or artificial membranes are fairly good insulator. This is because there are no free ions in the membrane, so there are no carrier proteins to carry charges. The specific conductance per unit area of pure bilipid or lipid bilayer is about G pure 10 to the power 13 per ohm per meter square. The conductance of biological membranes is much higher, usually by the several orders of magnitude even at rest. The reason is that there are different types of ion channels and other pores embedded in the membrane and allowing additional current to flow. When a current of 1 ampere passes through a section across which a voltage of 1 volt exists, then the conductance of that section is 1 Siemens. The Siemens is in fact equivalent to 1 ampere per volt. If G is the conductance of a component, R is the current through the component, and E is the voltage across the component, then G is equal to I over E. Conductance is inversely related to resistance. If R is the resistance of a component or device, then the conductance G is expressed as G is equal to 1 over R. This figure A showing the basic electrical properties of a biological membrane as electrical resistance, capacitance circuit or RC circuit. B, a more elaborated view showing the electrical elements necessary to carry currents underlying an action potential. Capacitance. What is capacitance? The cell membrane acts as an electrical capacitor. An electrical capacitor is a charge storing device which consists of two conducting plates separated by an insulating barrier. Because the lipid bilayer of the plasma membrane forms an insulating barrier separating the electrically conductive cellular solutions of the intracellular and extracellular side medium, the plasma membrane behaves as a capacitor. The capacitance is directly proportional to the area of the plates. Bigger the plates, they can store more charge, and it is inversely proportional to the distance separating the two plates. Capacitance also depends on the characteristics of the insulating material between the plates, which is the lipid of the plasma membrane in the cell. The unit of capacitance is farad. The capacitance of a capacitor is symbolized by C and related to voltage by C is equal to Q divided by V, where Q is the charge. If 1 farad capacitor can store 1 coulombs of charge when hooked up to a 1 volt battery. Biological membrane have a capacitance of approximately 10 to the power minus 6 farad, that is 1 micron or 1 microfarad per centimeter square of the membrane area. In the absence of integral membrane proteins, cell membranes possess electrical properties to those exhibited by parallel plate capacitors. Both plasma membranes and parallel plate capacitors are made up of a non-conductive medium that separates two conductive materials. In case of a plasma membrane, the non-conductive medium is the hydrophobic core of the membrane. Thus, the lipid bilayer can separate charge Q between the internal and the external conductive media, and this charge separation produces an electrical potential difference across the lipid bilayer that is termed as membrane potential or Vm. The figure is showing the membrane potential equivalent to a voltage difference across the plate of a capacitor. The cell membranes forms an insulating barrier that acts like a parallel plate capacitor. The capacitance of the membrane is 1 microfarad per centimeter square. Electrical potential across the cell membrane. Because of the unequal concentration of the ion across the cell membrane, the membrane has an electrical charge inside negative. Changes occur in the membrane potential to elicit action potential and give the cell the ability to send the messages to different parts of the body. In some cells, such as nerves and muscle cells, they are capable of generating quickly changing electrochemical impulses at their membranes, and these impulses are used to transmit the signals along the nerve or muscle membranes. What are the electrical potentials? Electrical potential across the cell membranes. First one is the action potential. Action potential are brief, rapid, large changes in the membrane potential in which the potential actually reverses. They are actually initiated as a result of spread from graded potential, and the graded potential are local changes in the potential that can vary in magnitude depending upon the strength and the duration of the stimulus. Stronger the stimulus, the more channels open. The larger the magnitude of the graded potential. The longer the stimulus, the longer channels may stay open and longer the graded potential lasts. Ion moments and the action potential. The whole basis of the action potential is based around a change in permeability to sodium and potassium due to the opening or closing of the voltage gated ion channels. Two discrete states, open meaning conducting and close, that is non-conducting state. Some channels also have inactivated state, open but non-conducting. The action potential is all or none, meaning either it is non-triggered at all, example too little depolarization, or the membrane is refractory, or it is triggered completely. Action potential. The resting membrane potential is a result of different concentrations of sodium ions and potassium ions inside and outside the cell membrane. The nerve impulses causes sodium ion to enter the cell, resulting in depolarization. At the peak action potential, potassium channels open and the cell becomes hyperpolarized. This figure is showing the different stages of the action potential. First one is resting state, second depolarization, third is rising phase of the action potential, fourth one is falling phase of action potential. And the rising phase, in this phase sodium channels are open, and in falling phase potassium channels are open. Course of action potential. Here I am going to tell more about the course of action potential, how it happens. First, there is rapid depolarization. During this, when partial depolarization reaches the activation threshold, voltage gated sodium channels open and then sodium ions rushed in. The membrane potential changes from -70 mV to +40 mV. Next phase is repolarization. The sodium ions close and become refractory. Refractory meanings they become inactivated, or another impulse cannot initiate an action potential. Depolarization triggers opening of voltage gated potassium channels. When channels are open, potassium ions rushed into the cell, repolarizing and then hyperpolarizing the membrane. After hyperpolarization, there is resuming of the action potential occurs. At that time, potassium channel close, repolarization resets sodium channels, ion diffuse away from the area, sodium potassium transporter maintained polarization. The membrane is now ready to fire again. Transport across the cell membrane. First one is membrane permeability, that is, the membrane serves as both an insulator and a diffusion barrier to the movement of ions. The second, ionic basis of membrane potential. The most important ions are sodium, potassium, calcium and chloride. Next, involvement of ion channels. Two types of ion channels are mainly involved. First one is voltage gated sodium channels and the second one is voltage gated potassium channels. Selective permeability of cell membrane. First, what is permeability? Permeability, what is allowed to cross the membrane. Impermeable, meaning nothing gets through the membrane. For example, iron, glucose, amino acid cannot pass through the membrane. If membrane is permeable, anything gets through. Cell membranes are semi-permeable, meaning some molecules pass through freely, some must be transported across. As I told you before, lipid bilayer is selectively permeable. Some molecules cross easily or go through the channel, others need energy to get through. Coming to membrane permeability, all living cells maintain a potential difference across their membranes. This membrane potential is due to the difference in the concentration and permeability of the sodium and potassium on either side of the membrane. Ion pumps and channels are electrically equivalent to a set of batteries and resistors placed in the membrane. and therefore generate a voltage difference across the membrane. In principle, there is no difference between resting membrane potential and dynamic voltage range like action potential from a biophysical standpoint. All these events are caused by specific changes in the membrane permeability of potassium, sodium, calcium and chloride, which in turn result from the concerted alterations in the functional activity of diverse ion channels, transporters and exchangers. Ionic basis of membrane potentials. The most significant cation for the action potential are sodium and potassium. Both of these are monovalent cations that carry a positive charge. Action potential can also involve calcium ions, which is a divalent cation that carries 2 times positive charge as compared to these sodium and potassium. The chloride plays a negligible role in the action potential of most animals cells. Electrical signals within the biological organisms are in general determined by ions. Ion channels transport the ions across the cell membrane. Ion channels. Ion channels are the core of our electrical system. Channels are present in almost every cell. Ion channels are transmembrane proteins with a port through which ion can move between extracellular space and cell interior. There are hundreds of different ion channels and they are distinguished based upon their ion selectivity, gating mechanism and sequence similarity. Most ion channels are selective for one ionic species. Voltage-dependent ion channels play a fundamental role in generation and propagation of the nerve impulse and the cell homeostasis. Physiological functions of ion channels. Various functions are performed by the ion channels in the cell. They maintain cell resting potential, and electrical conduction of signals are mainly mediated by ion channels. Synaptic transmission, transfer of ions, metabolites between adjacent cells are by ion channels with the help of ion channels. Sensory perception, excitation contraction coupling, stimulation secretion couplings, these are the few functions performed by the ion channels. Types of ion channels. As I told you before, there are various types of ion channels. The ion channels can be simple pores like gap junctions or ligand-gated ion channels. For example, nicotinic receptors. Voltage-gated ion channels, mainly potassium channels, sodium channels and calcium channels. pH-gated, mechanically gated and pumps. Example, ATP synthase, sodium-potassium ATPase. Today I am going to talk more about voltage-gated ion channels. These are the cationic channels, sodium channels and potassium channels. These two channels are responsible for the action potential in the nerve cells or neuronal cells. Gated ion channels. General information about the channels. They have pores in the membrane that open and close in a regulated manner and allow passage of ions. They are passive transporters, meaning ion flow from higher to lower concentration. No energy is used. If there is no gradient, ions will not flow. Small, highly selective pores in the membranes move ion or water. In ion channels, the rate of transport is very fast. If you compare them with the ATPases or the pumps in the membrane, the rate of transport of ion is 10 to the power 7 ions per second. If you compare it to sodium potassium ATPase, that is a pump in the membrane, the rate of transport of ions is 5 into 10 to the power 12 ions per second, meaning you can say there is a huge difference in the rate of transport between these two voltage ion channels and the pumps. Transport is always down the gradients and cannot be coupled to energy source. Gating mechanism. What is gating? Gating is nothing but opening and closing of an ion channels. What gates the ion channels? Non-gated means they are always open. Gated means voltage change across the cell membrane, open or close the ion channels. It can be ligand gated or mechanical stimulus or thermal fluctuation can gate or open the channel. Two discrete state of ion channels, open and closed. Opening means conducting state. When channel opens, an ion passes through the channel. The closed state, it can be non-conducting. Some channels have also inactivity state, open but non-conducting. In this state, channel have to go to inactivity state before going to open again. Part of the ion channel structure or external particle block, otherwise channel is open. The conformational change in the membrane protein are responsible for opening and the closing of the pore, which I am going to tell in detail in the next couple of slides. Energy to switch the channel protein from one conformational shape or structure to another comes from the gating sources. Voltage gated ion channels, as their name indicate, they are voltage gated, meaning they open in response to change in membrane potential or membrane voltage. Subsequently open and inactivate. Opening, some channels open for longer duration, some open for shorter duration. They are specific for a particular ions, means sodium channels is highly specific for sodium, even if the potassium, which has the same amount of charge, both of them have one positive charge on them, but sodium channel is specific for sodium and it will only pass the sodium ions. They share common domain structures. They are regulated by external signals. And these two channels, sodium channels and potassium channels, they are mainly responsible for action potential in the neurons. In cardiac myocytes, calcium channels are also involved. Calcium channels are also involved in secretion, signaling, muscle contraction and gene expression. Now coming to the structure of the voltage gated ion channels, all these cationic channels, meaning sodium channels and potassium channel, contain 4 subunits, each containing 6 transmembrane segments. Potassium channel is a tetramer, meaning it is made up of 4 alpha subunits. Sodium and calcium ion channels have 4 peptides connected into a chain. All these channel proteins have 3 basic parts. One is voltage sensor, the second one is a pore or conducting pathway, and third one is the selectivity filter. Sodium channels. Sodium channels are one large polypeptide of 4 domains. They are responsible for action potential initiation and propagation in excitable cells, including nerve, muscle, and neuroendocrine cell types. They are also expressed at low levels in non-excitable cells where their physiological role is unclear. Sodium channels are the founding members of the ion channel superfamily in the terms of their discovery as a protein and determination of their amino acid sequence. Opening and closing or opening of voltage sodium channels result in influx of sodium into the cell when sodium comes into the membrane and their current is always coming into the cell. Sodium channel structure. The figure is showing the structure of the sodium channels. As I told you before, it has 4 transmembrane domains, domain 1, 2, 3 and 4, and each domain has 6 transmembrane segments. Transmembrane segment 4, it is main voltage sensor, meaning it senses the voltage or any change in the membrane potential, and it has positively charged amino acids. And there is a P-loop between segment 4 and 5, and all these P-loops come together and make the pore in the membrane. Sodium channels, gating and permeation. As I told you before about gating, the gating is nothing but opening and closing of ion channels, and permeation is how the channel pass through an open channel. This diagram is showing voltage gated ion channels, how they open and close. They are closed at the resting membrane potential. At rest, the neuronal membrane has membrane potential of minus 70 millivolts. When there is any stimulus or impulse, they open in response to a nerve impulse, and the gate opens and sodium enters into the cell. And they open for a shorter duration. Their current is very short-lived. They inactivated for a brief period following activation. The channel does not open in response to a new signal. As I told you before, when action potential comes, the sodium channel becomes refractory, meaning they are inactivated and cannot be opened unless they are closed. Potassium channels. Potassium channels, voltage-gated potassium channels of vertebrates typically are tetramer of 4 identical subunits arranged in a ring, each contributing to the wall of the transmembrane potassium pore, each subunit containing 6 transmembrane segments. The majority of potassium channels are characterized by 1000:1 selectivity ratio for potassium over sodium. Though potassium and sodium ions have the similar charge and vary only slightly in their radius. Voltage-gated potassium channels are inactivated by membrane depolarization. Upon depolarization, these channels open, allowing the efflux of potassium from the cell down its electrochemical gradient. They function in the heart and brain to repolarize the membrane potential and to terminate an action potential. Now coming to calcium channels. Calcium channels, they are one large polypeptide of 4 domains. Opening of voltage-gated calcium channel results in influx of calcium ion into the cell. This influx of calcium plays an important role in excitation-contraction coupling in cardiac and skeletal muscle cells. The reversal potential for calcium is extremely positive, so calcium current is almost always inward, resulting in an action potential plateau in many excitable cells. In neurons, mostly responsible for the entry of calcium into the presynaptic ending following depolarization and subsequent endocytosis of neurotransmitter. Circuit diagram of neuronal membrane based on Hodgkin and Huxley model. The figure 1 is showing the passive current flow in a neuron can be modeled using electrical equivalence circuit. Figure B is showing the circuit includes elements representing the ion selective membrane channels and the short circuit pathways provided by the cytoplasm and extracellular fluid. Membrane impedance. Impedance, denoted at capital Z, is an expression of the opposition that an electronic circuit component or a system offers to alternating current and or direct electric current. The impedance and the potential measurements have been made on several artificial membranes. Impedance changes are determined as a function of current and of the composition of the environmental solutions. The difference between resistance and impedance is that resistance is a concept used for direct current, whereas impedance is the AC equivalent. The resistance is due to electrons in the conductor colliding within the ionic lattice of the conductor, meaning that electrical energy is converted into heat. Different materials have different resistivities. However, when considering AC, one must remember that it oscillates as a sine wave, so that sine is always changing. This means that other effects need to be considered, namely inductance and capacitance. To summarize whatever we have learned so far, so in summary, I can say all living cells, not just excitable cells, have resting membrane potential. The electrical properties of the cell membrane are important in determining how electrical signals spread along the plasma membrane. One of the typical features of a cell is that it regulates the transport of electrically charged ions across the lipid bilayer and therefore the electrical potential across the cell membrane. The cell membrane possesses electrical properties to those exhibited by parallel plate capacitor. And last, impedance is an expression of the opposition that an electronic component, circuit or system offers to alternating and or direct electrical current. Thank you.